So up next for our tools, we have Brainome. So Rand's going to be presenting on some of the work associated with Brainome and how it fits into BICCN. So with that, go ahead.
Great, thank you, Joe, and thanks for the invitation. As I was saying, this is a fun opportunity to talk about, in a really practical way about kind of the tools that we've tried to develop around the BICCN data. So I'm happy to keep this quite informal. So please feel free to shout out any questions as they come up. My goal here is to give everybody an overview of the tools we've built for accessing and interacting with data, so that you can go ahead and use them yourselves. So actually, let me share my full screen. So I'm going to start with my slides, but then at some point I'll transition to actually a demo of the website. So as Joe said, I'm Aran Mukamel, I'm at UCSD, and I've been very involved with the mouse brain single cell omics working group within the BICCN, whose goal has been to really generate an atlas of brain cell types across the mouse brain. And so this was the first goal of the brain initiative, was to define a parts list for the brain. I like to compare the biological parts list of the brain with what we think about in terms of artificial neural networks. AI and machine learning have been so revolutionary the last few years, but if we think about the components that the typical neural networks are built out of, they're relatively homogeneous. They're far less diverse than the parts list that we can define in the brain. And so, as I said, goal number one of the Brain Initiative was to define cell types. And I think for this audience, I probably don't need to dwell on this too much, but having a reference of cell types will really enable progress across experimental and conceptual neuroscience. So what I want to tell you about today are the tools that we've developed for interacting with multimodal data. And I think multimodal interaction and integration is really essential because by looking at cell types across different modalities, in particular transcriptomic and epigenomic types of data, we can get independent and mutually cross-validating perspectives on the cell types. So for example, if there's a cell, that has one particular gene that seems to be expressed compared to other cells, sometimes it's unclear how to interpret that and whether that's really a genuine stable difference in cell type, or is it more of a transient cell state reflecting, for example, activity-dependent gene regulation, could be circadian rhythms. Many things can affect gene expression on various timescales. Whereas when we see signatures that are consistent across, for example, DNA methylation, chromatin accessibility, and transcriptome, we start to have more of a confident sense of a functional cell type. Of course, we eventually want to also connect with other types of anatomical, morphological, and physiological data. But for today, I'm gonna focus on the molecular data, and I'm going to be focusing on a data set. So I'm going to demonstrate our tools using a data set that comes from the first phase of the BICCN, which focused on the mouse primary motor cortex. So I'll very briefly introduce the data set. So this is showing you where the mouse MOP is. And in terms of the data that was brought together for this effort, there were over 600,000 cells across 9 data sets, and these really spanned from different types of transcriptomic data. So here you can see single cell RNA-seq, single nuclei RNA-seq using SMART-seq and 10x, as well as open chromatin using SNA-TAC-seq and DNA methylation. And I'm not going to have time, I think, to go into any of these methods, but feel free to ask me any questions if you have questions about the interpretation and the meaning of these data sets. But for now, I think it's enough to just point out how complementary they are. So for example, some of the data sets really spanned, had a massive scale with hundreds of thousands of individual cells profiled, especially the 10x RNA-seq, as well as the ATAC-seq data. But those modalities tended to profile a relatively modest number of molecules or unique DNA or RNA fragments per cell. Whereas other modalities like methylation, SNMC-seq, and SMART-seq transcriptomes profile many, many more, millions of transcripts or millions of molecules per cell, but in fewer cells. So I'll breeze through this, just showing that we're able to do an integration. So this is analysis of each modality on its own, showing cluster analysis, the cell types that can be defined. And then we really focused on multimodality integration. So the consortium took, as a collaboration, took a couple of different approaches. My lab contributed a method we developed called single cell fusion. Josh Welch and Evan McCosco developed a complementary method called Liger, and both were able to successfully integrate these data. So here you can see an example of a browser shot, and I'll go into much more detail about this, but showing 4 different types of data. So we've got DNA methylation in green, ATAC-seq in blue, single cell RNA-seq in purple, and single nuclei RNA-seq in red. And this is not all of the modalities that we had in BICCN, but these are the 4 that we've chosen to represent in this browser view. And what you can see here is at this locus, the TAC1 locus, you can see low levels of DNA methylation, high levels of chromatin accessibility, and high levels of gene expression. And all 4 of these tracks come from a single GABAergic neuron type, the parvalbumin expressing, relin and calbindin-1 expressing MGE-derived GABAergic neurons. So that's a mouthful, but what you can see is these correspondences. So, and these are the correspondences that we use to link the signatures across modalities and to solve this, this data integration problem. This is just, again, I want to breeze through this. This is just a low dimensional embedding. In this case, it's a UMAP embedding, showing each of the 600,000 cells colored by the modality that it came from or by the cell type. And this is just showing that we can integrate all of the cells into a joint space. Just to convince you that the signatures really do correspond to each other across modalities, here you can see, I'm just showing the excitatory neurons in this case. So organized from superficial layers, layer 2-3 IT cells through layer 6 and IT cells, and then we've got layer 6B, layer 5 PT neurons, layer 5 near projecting cells, etc. And at this particular locus of the TSHZ2 gene, you can see a really dramatic signature of low DNA methylation throughout the kind of the first intron of the gene. And this is a common DNA methylation signature of a very cell type specific active gene. Specifically in these 2 cell types, in the layer 5 near projecting neurons, 2 different clusters of layer 5 near projecting neurons. Now, what you also see is in this very same cell types, you see high chromatin accessibility and high gene expression. And so this is an example where we can, see in the browser the specific correspondence between cell types. And I'll get into more detail on this a little bit later, but the usefulness of looking at this in the genome browser, as opposed to other kinds of ways that you might represent the data, is that this really gives you a rich ability to see exactly where the different signatures are. So for example, the DNA methylation here is represented with single base resolution. Each of these green ticks is a single cytosine, a CG position, and the height of the tick is the amount of methylation there. So you're able to see with single base resolution, for example, we can look here at this region that seems to have low methylation across cell types, and we can look at whether there's an open chromatin signature specifically at that location as well. You can also represent the data in other ways, for example, on the UMAP. So this is just showing the overall gene expression level across each of the 4 different transcriptomic data sets. And you can see these 2 clusters are expressing TSHZ2. And then the same 2 clusters also have a high level of chromatin accessibility and low level of DNA methylation. And we can do this for other marker genes as well. Okay, so I've labeled this here single neuron analyzer. Single neuron analyzer is another name that we've used. The website that this is hosted at is Brainome. So I think for today we can just use Brainome as the name. And our goal is to be able to analyze millions of cells across these different modalities collected from multiple sites. Ultimately, we'd like this to be a tool for iteratively and collaboratively and analyzing data sets. So for example, we might have expertise in looking at the DNA methylation signatures, but we want folks who are more expert in the RNA-seq, for example, to be able to compare their data with ours and refine the annotations. And we want this to be, accessible so that people can interact with it over the web and not have to download data necessarily to analyze it. And we also, are interested in working closely with the other members of the data ecosystem, NEMO and BCDC, and the Broad Terra folks who we collaborate with to develop some of the pipelines. Okay, so I want to skip this in the interest of time. Let me skip a couple of slides ahead and go into, yeah, I don't think I need to talk about the our method for data integration. So what I'd like to do now is really just show you some screenshots of the browser to tell you a little bit about how it works. And again, I really, if there are any questions, please do stop me and speak up, because this is meant to be, a practical kind of thing. So here's the URL. Actually, let me click the URL and just show you that it works. So if you go to brainome.ucsd.edu/BICCN-MLP, it'll redirect here. So this is a base resolution epigenome browser. We can zoom in a little bit. So let me type in the name of a gene here. So for example, we're looking now at GABAergic neurons. So let me go to the gene LHX6. It can search transcripts, and you can select which transcript you want to look at. It'll take you to the start site of that gene, centered at the middle of the screen here. We can zoom in a little bit with this magnifying glass to different levels of resolution. So if I zoom in, you can really appreciate that this is single base resolution data. Let me make the tracks a little bit larger. So these arrows here allow me to change the height of the tracks. You can also do it for individual tracks if you want, by selecting here the resize feature. So for example, I can make this track bigger or smaller. I can do that for each of the tracks. Let me go back to browsing mode. And so in browsing mode, I'm able to just click and drag left and right. Let me make the tracks bigger again. So again, each of the ticks here is one CG position. And the height of the tick is the amount of methylation that's measured there. So here we're looking at the promoter of the LHX6 gene, and you can see all of the cell types have low DNA methylation at the promoter. So this is a very common thing. If we zoom out and look at maybe some other promoters, you can see most promoters are unmethylated. So this is a promoter actually that's shared between two genes on the two strands, very low DNA methylation, and that's really a common feature across genes. But so we can see that here also if I put my cursor over this promoter. CG methylation levels throughout the rest of the genome tend to be high, so you can see almost everywhere else these ticks go all the way up. But within the gene body of LHX6, you can see this really interesting pattern where specific cell types, in particular, this is a marker of MGE-derived GABAergic neurons. So you can see that SST neurons and the parvalbumin-expressing neurons have very low levels of DNA methylation throughout the whole gene body, not just at the promoter, but extending throughout the whole gene body. Whereas the CGE-derived GABAergic neurons like VIP cells, SNCG, and these LAMP5 neurons have higher levels of DNA methylation. Now, we also have tracks in here. These black tracks are showing regions where we've statistically called differences in methylation, so in particular low methylation, indicating active enhancers. And I believe, let me see. So there's lots of other tracks that can be turned on and off, and I'll show you some other methods of doing that in a minute. But before we get into that, let me scroll down here and look at the attack-seq data. So we also are showing on the same screen the attack-seq tracks from the same cells in the same order. And what you see is the same cell types that have low DNA methylation, such as SST-CALB2, SST-MYH8, also have high levels of attack-seq open chromatin, again, at the promoter and throughout the gene body. Now, what I'd like to now, so one thing I want to point out, this is based on, this genome browser is a little bit different from other browsers you might be used to, like the UCSC browser. It's called NOJ, and it was developed by Julian Tanto Filippini starting around 2007, and further refined by some of these folks, Swaming Chen most recently, and we've contributed some code to refining it. But we've also then built this tooling around the epigenome browser that should allow you to interact with it in other ways. So for example, we can choose which cell types we want to show. So for example, if you want to show excitatory neurons instead of the inhibitory cells, I can select excitatory neurons, and then I have to click reload browser. And it's going to refresh this frame. And now I've updated the tracks to include all of the different excitatory neuron types. Now with the excitatory neurons, LHX6 is not as interesting, right? Because this gene is basically highly methylated in all of these cell types. But if I go to another, does anybody have a favorite gene? Anybody want to nominate a gene for me to go look at? Make this a little bit interactive. This is a test to see who's awake. If not, I'm going to have to choose a gene.
We have Prox1 in the chat.
Yep.
All right. Great. Thanks. Perfect. Prox1. Let's take a look. Now, Prox1 might be more interesting if I look at the GABAergic neurons. So actually, let me go back to the GABAergic neurons. So inhibitory neurons. And it'll keep the same gene that we were at, Prox1. So here's Prox1. And so now, as you know, Prox1 is a marker for CGE-derived neurons. In particular, you can see the VIP cells have low DNA methylation, as well as SNCG and LEM5 neurons. Let's see. So that allows you to choose which cell types you're displaying. You can also choose individual cell types, and there's two ways of doing that. So one way is to just display the whole list, and this is searchable. So if I, for example, if I want to show, let's say I want to show the layer four neurons, this shows them to me, and I can select, okay, I want to show MCG, MCA, ataxic, maybe all of these for these different clusters. And then when I'm done, I can again reload the browser, and it will add those tracks to the list of tracks that are shown. Another thing you can do, and something to point out here, so now I've added non-CG methylation. So for example, MCA is a feature of methylation that's unique to neurons. We don't really find it in non-neuronal cell types. And so it's shown in a different color. It can be sometimes hard to see which track is which. So when you hover over the track, it'll give you the name of the track. You can also reorder the tracks. So for example, if I want to move one of the tracks down, I can move them up and down in this list. You can turn them on or off by double clicking them. So for example, I can double click a track and it will disappear. If I want to bring it back, I can bring it back. So here's another way to interact with the track list. The tracks are all listed down here. So for example, if I want to add an ataxic track, here is the list of all of the inactive tracks. And again, I can double click one of the tracks, and it will, by default, it will add it to the end of the list down here. Okay. One thing that I think is really important is to be able to share and reproduce these browser shots. So of course you can take a screenshot, but another handy thing is to copy a shareable link. So if you click copy the link, and then you can send this link to a friend, and it should reproduce more or less the configuration of the browser that you were looking at, with the same track list, the same locus that you were looking at, etc. Okay, so if I go back to let me show you now a couple of other ways of configuring it. So for example, let's say I want to look at excitatory neurons, and now I want to choose which modalities to show. So for example, suppose I want to look at MCG, ATAC, scRNA, and snRNA. I can then reload the browser. And this might actually be too many tracks to show on one page, so maybe I'll reduce the list a little bit, but by default, they're grouped by modality. So you'll see first the MCG, the methylation tracks, then we see the ataxic tracks. Oh, and again, we're looking at the wrong gene. So let me go to like maybe the ROR-B gene, which should be specifically active in layer 4, middle layer 4 and 5 excitatory neurons. You can see again, lower DNA methylation in these layer 4, 5 neurons, DMRs or enhancers that are being called in those regions, attack-seek accessibility, and then gene expression. It's sometimes useful to be able to change the scale within these tracks. So you can do that here with these arrows. So we can make the tracks, smaller. We don't, unfortunately, have an auto scale feature. So if you're used to like the IGV browser, we don't have an auto scale feature. I actually think sometimes it's handy to be able to really control the scale in this way. And all of the tracks are kind of group scaled, so they're all scaled in the same way. Let me see. So here I'm showing almost all of the data. Oh, one other thing I want to show is another way of grouping the cells. So let me switch to a smaller group of neurons. Let's look at just the MGE derived neurons. And I'll go to an interesting gene for the MGE cells, maybe that TAC1 gene that I showed you earlier, tachykinin. So again, this is specific to just the parvalbumin expressing cells. Now it's interesting. So here you can see high levels of expression in the parvalbumin expressing cells. There's also somewhat lower expression in these other cell types. To appreciate that better, I'm going to turn down the scale of the tracks on the RNA, and so you can really see that the expression is really much higher in these in the PV cells. And then I'll turn back up the scale on the attack-seek track, and you can appreciate that as well. Now, one thing I want to show you is that it's sometimes handy to be able to group the tracks in a different way. So let me group them now by cell type and reload the browser. And what this will do is now I've got each of these groups of tracks corresponds to one cell type. So for example, let me turn down the height on the attack-seek track. So now you can see this, I'll turn up the height on the RNA. Okay, so what you can see is that again, these p-valgurelin expressing, CALB1 expressing neurons have low DNA methylation, high chromatin accessibility, and high gene expression. And then when we compare head to head with other MGE-derived GABAergic neurons, you can see that the methylation level is much higher. The attack-seek is much lower, and the gene is not really expressed apart from a very low level in other cell types. Now, one thing I want to point out. So far, I've been focused on gene-centric features, but if we look here upstream of the TAC1 promoter, so this is so you can see here the cursor shows me the exact genomic coordinates. So here I'm looking at chromosome 6. By the way, you can type in coordinates directly if you want. So, but here I'm looking at chromosome 6 at position 7,554,000 base pairs. Over here, I'm at 7,529,000. So, a few tens of kb upstream of the gene promoter, and what you see is an enhancer that's marked by a peak of attack-seek, a peak of open chromatin, low level of DNA methylation. It's got this enhancer call here. We could zoom in on it, and this is important because this is what really the value added of the epigenomic data. Obviously the RNA-seq is not giving us any information about this putative enhancer or regulatory region here, and these are the kinds of regions that we're often interested in for finding cell type specific regulation. And if we zoom out and look at other regions, we can see other potential regions throughout this 100 kb region around the gene that might also be potential regulatory elements. Now, one kind of open biological question is we don't know whether these enhancers are regulating this gene, although it stands to reason here. They're quite close, and there's not another gene nearby. But that's one of the biological questions we're interested in working on is how do we link these enhancers with the genes that they regulate. If we scroll around, you can see other examples of genes. So for example, here, this gene looks like it has a very strong open chromatin signature throughout the gene body. You can see what other intergenic enhancers look like as well. So are there any questions about this epigenome browser before I move on a little bit? Okay, so another thing that we have is, so we have link outs to other tools. I'll just show here, we have links to the BCDC, Nemo Analytics, and all of these are complementary. So there are, lots of other ways of viewing complementary data sets. Our tool includes also this other way of looking at the data we call the cell browser. So let me preview that here. So the cell browser, this is something we developed, although there's now a number of other tools that do, that do this as well, and maybe a little bit faster. So it's similar to cell by gene, to the UCSC single cell browser. But let me just show you how this works here. So we've got a UMAP embedding, or in this case, a t-SNE embedding of the cells colored by cell type and also by methylation level. In this case, this is the non-CG methylation level. So you can see here, we're looking at the gene GAD2. The gene body methylation is low in these cells. So these are likely the inhibitory neurons. If we open the menu here on the left, oops. This is always the danger of doing a live demo. You have to hope that everything works. There we go. It'll open some controls for you. And so, for example, you can type in a different gene if we want to look at PROX1, somebody said was interesting. We can type in PROX1 and update the browser. And what this should do is update the coloring on the right here to show the methylation level of the PROX1 gene. And so now you can see it's specifically marking these CGE-derived neurons. We can also pull up a box plot that shows, box and violin plots of the distribution of methylation for each of the clusters. Now, these are clusters that are just labeled 0 through 28. We can also group them by an annotation, and so this should give more meaningful names to the clusters. So you can see low levels of DNA methylation at these CGE-derived neurons. For example, LAMP5 cells. These are VIP cells, and another cluster of VIP cells.
Do we want to take a couple seconds for questions?
Oh, yes,
and
yes.
Yang can get set up.
Perfect, thank you. Yes, so are there any questions?